by Dr. J.Jeyakanthan sir on the topic Journey from Cells to Atom, Integrating Multidisciplinary Research Approaches. Our department science club named as Triple Helix, honoring the great biophysicist Professor G.N. Ramachandran sir. The name Triple Helix is intrigued, and this is where the starting point for us to learn transdisciplinary areas. Understanding the hierarchical organization of biological structure requires a multidisciplinary approach and atomic level knowledge. This can reveal new insights into complex biological systems. Atomic level understanding can inform designs of new materials and technologies. In this session, Dr. J. Jeyakanthan sir will enlighten more about multidisciplinary research approaches. We are eagerly waiting to learn from you, sir. Now, I would like to call upon Hema Malini of 3rd BSc Physics to introduce our speaker.
Good morning. On behalf of my department, I am esteemed to introduce our today's speaker, Dr. J. Jeyakanthan. Sir, graduated in physics and obtained his PhD in crystallography and biophysics from the University of Madras. His research based in the broad subject of structural biology, biocomputing. His area of specialization is small and macromolecule X-ray crystallography. He conducted his postdoctoral research under the supervision of Professor M. Vijayan in IISc Bangalore. Later, Sir worked as a research scientist for more than a decade in Riken Harima Institute, Springate, Japan. Dr. J. Jeyakanthan has more than a decade of teaching and research experience in structural biology, biocomputing, and computer-aided drug design. His research focuses on the structural and functional aspects of disease-associated protein targets, which shares high similarities with human genome. Dr. J. Jeyakanthan sir has significantly contributed to the understanding of structure of therapeutic protein targets in PAC1-mediated cancer and SART4-induced type 2 diabetics, and also works on structural analysis of NSP2 protein in SARS-CoV-2 along with phototherapeutic agents and synthetic drugs. Dr. J. Jeyakanthan sir has visited more than 15 countries to deliver lectures and for collaborative research purposes. And sir served as a visiting scientist in National Synchrotron Radiation Research Center, Osaka University, Taiwan. Sir, I am feeling proud to talk about your achievements. Dr. J. Jeyakanthan sir has received several awards, especially the UGC Research Award based on his research performance in structural biology, biocomputing, and bioinformatics in the year 2016, and Governor Sir nominated him as a syndicate member in universities, and the Tamil Nadu State Council of Science and Technology awarded him the Tamil Nadu State Award in Biological Sciences in the year 2018. And Sir Research Fellowship CSIR in December 2000 and UNESCO in 1999. Sir also received DST India Young Scientist Award during his PhD in 1999, MHRD Leap Award by NIT Tiruchi and NTU Singapore in the year 2019. Leadership Development of Dean of Faculty and Head of the Department of Universities, National Institute of Education Planning and Administration, New Delhi, India in the year 2024. Sir research contribution have earned him a place in several prestigious committees in the Government of India and abroad. He is an active member in academic and curriculum development in various universities and institutions across India. Dr. J. Jeyakanthan sir also holds a life membership in 9 scientific professional bodies, namely the American Crystallographic Association, Bioinformatics and Drug Discovery Society, British Crystallographic Association, Indio Crystallographic Association, and World Directory of Crystallographers. Sir is an explorer in science and a true architect of future discoveries. Sir, thank you for being here to guide us. You are an inspiration to all of us. Now, we are looking forward for your valuable insights. Now, the session is over to you, sir. Welcome, sir.
Very good morning to all once again. So, I really thanks to once again to Madam and the student to tell about me. So, here I am going to talk about the journey gels to atom integrating the multidisciplinary research approach. So you are all from physics. I think you may be easy to understand because I am completely from physics, change it to the biological science. Next, further. Yes. Maybe so you are all know about this organisms. So organisms is you tell about the what are things are there in our body. You just imagine. So many organs, right? Many organs. The many organs is made up of what? Tissues, right? Tissues made up of cells. Cells made up of molecules, right? DNA, protein, and everything. And molecules made up of atoms. So here we need some techniques. What kind of techniques do we need to identify? Physics, na? X-ray, right? The X-ray is the ultimate method to identify the atoms and molecules and everything. See here you can think about that. So in our body there is a millions of there is a 23,000 genes are available in each cell. You can think about that. Our body is made up of huge cells, right? The each gene will do a different kind of function, any kind of function. So you just think about that. If you take any kind of one organ, there is a n number of proteins are there, like 600, 700 proteins are there. Then each protein kind of. So we need to identify the each protein function. Then only we can go for the to identify the drug molecule for to cure the disease. That is the very important. So you can imagine. So each cell there is a 23,000 genes are there. The each gene is a one protein. So doing a one kind of function. So how we can to solve that protein structure? That is the very concept. Here we are using the physics technology from the biological to the let's see. So as I mentioned, the human. From there we are taking tissues on a human cell. Then from the cell we are getting the chromosome. From the chromosome we are getting the DNA. From the DNA we are getting the mRNA. Then we are getting the genetic codes, right? Adenine, thymine. Maybe you heard about that. From there we are getting the amino acid. From there we are getting the amino acid. There is a 20 amino acid. There is a 20 amino acids playing a major role in our body. There is a combination of 20 amino acids. There is a one gene or one protein, you can say that. So where we are getting the amino acid, you can think about that. The obtained from nutrition. There is a essential amino acid. There is a 20 amino acid only. Essential amino acid and non-essential amino acid where we are getting from our body is synthesizing. So the combination of 20 amino acids is playing a major role in our biological system. So we need to identify that amino acid, that structure of the amino acid or structure of the protein. So why we want to study now? Because once we identify the protein, that protein is a macromolecule. Then there is a small molecule, that is a drug molecule. Once the protein we have solved structure, then small molecule, then we can make a complex. That means the docking or binding. Once binding to the protein, the mechanism will start. So that is why we need to study about the proteins. So this is the method we are taking care. At present, there is a 230,000 structures from the various organisms. There are many organisms are there, not only human. Bacteria is there, fungi is there, many organisms are there. That must be the all whole genome sequence is matched with the human because we cannot identify a drug for the directly for human. We have to go for the other source, other organism, that is a bacteria. Once we identify the drug, that will be implemented to the human genome. That's why there is a human genome project, mapping with the human genome. So we needed to target first other organism to solve the structure of the protein, then we need to make it to the human genome, right? Okay, next. So there is a whole genome sequence and annotation studies. Why we want this study? Because, so you think about that, next slide. So we are hospitalized So we are in the ICU unit. Okay, we are in the ICU unit. We are collecting the culture sample from us because we went for some kind of disease like maybe urinary tract infection or something like that. So we are going for the hospital. They are collecting the sample and testing the whether which is the antibiotic drug we need to give for the patient, right? So for that we needed some methods. For that we can see here method sample collection from the patient. So first we are collecting the sample from the patient. Then we are doing all kind of already existing antibiotic drugs. We are testing first. Once we collected the sample, our urinary sample we are taking from the patient. Then using the already available antibiotic testing. Then once tested, we need to check about what kind of infection caused. There is a bacterial infection. That is the very, very important. You can think about that. Then we need to get a DNA. That is very important. From the culture, we are getting the DNA isolation. From the DNA isolation, we need to get a 16S RNA sequencing. From there only we can identify what kind of bacteria affected us. So that is very important. So once we know which kind of bacteria, then based on that, the doctor is giving the medicine for the antibiotic drug. So this is the way we need to identify. Then we are doing the whole genome sequencing. That means, so suppose collecting each person there is a whole genome sequence is there. If you take one kind of disease related, from the culture, we will get the whole genome sequence. That is a DNA sequencing. From there, we can get the amino acid sequences, like DNA sequences. Then it will give the n number of, like many n number of things are like small segment. That segment we need to connect. Then use everything like this kind of method. Finally, we will get the annotation for phage identification. Finally, we will go for the what kind of the disease caused in the your culture oriented. Then from that we can take, this is the methodology. Once you are collecting the sample from the patient, we need to do this kind of things. Why we want to do, that is the very important. Okay, next. You see here, so we are collecting sample and we are doing all kind of things. So why we want to do? Because, so for example, we are all staying here. That is, all are maybe affected by the some diabetes related disease, right? For example, I am telling. So here already existing medicine is causing maybe the 100 persons are affected by diabetics, maybe 8 persons skewed by the already existing medicine for cure the diabetes. Remaining 20% is not cured. How? Why? What is the purpose? That means here we need to think about that. We cannot change the drug, right? Immediately we cannot change the drug. Then what we can do? So we need to collect a sample that remaining 20%. Then mapping with the already 80%. Then the genome sequencing where somewhere maybe missed or something, or somewhere maybe replaced some other codon or amino acid. So what we can do here, then we cannot change the genome sequences. We can think about that the already existing drug. We can go and change some functional group in the drug. Then change the functional group. Instead of nitrogen, we can add some oxygen. Then make a new drug. Then give to that drug to the that concerned persons. Then it may be cure. Cure means what? That may be control. The control the disease. So for this purpose only we need to collect the samples from each patient or each person. Then we need to make a whole genome sequences. Based on that, we can go for the identify the drug for the particular diseases. Okay, so this is the method to use all the bioinformatics tools. Once we collected sample, getting all the sequences, use the bioinformatics tool, you will get finally that is a sequence of our each person. Okay, next. So like you can see, this is the method. Like this is the method, we are getting the DNA fragments like segment. Then we use this kind of instruments to make all combined together. Finally, we will get this kind of genome sequences. This everything is being by the bioinformatics. That is a next generation sequencing workflow. So once we are collecting sample, you can think about that only we are collecting water oriented, like water oriented, urinary. Only urine sample we are collecting. Finally, we are getting the genome sequences. You can think about that. What are things are there? Okay. Next. So use this kind of technology. Finally, we can do all kind of bioinformatics tools. Finally, we can get the proteins and RNAs in the sample. This is a sample, like liquid sample. Still, I am not coming to this kind of structure. So we need to go for next. So we need to do all kind of analysis, whether this sample is affected any kind of antimicrobial resistant genes, multi-locus sequencing, this kind of function we need to identify in the culture. So based on the using the bioinformatics tools. Next. So once we got the all information, finally we need to go on to submit the data, the sequence data. There is a NCBI. Maybe you can look at the website. You can get the all kind of genome sequences. Each person which is affected with any kind of diseases, they will submit. But it costs very high. So each person, we are going to the hospital, they are collecting sample. We need to send that sample to the for sequencing. Maybe doctor will say about that, you will wait for 3 days to get the report from there, right? For that, so that means they are identifying what kind of bacteria is affecting for your urinary tract infection. So based on that only they will give the antibiotic drug. So once we got all the sequences, we needed to submit in these databases. From there, we can take here millions of genome sequences are there. The student, you can do your project. Use this website. You can get some kind of protein sequences, genome sequences. Only computers enough. Everything is freely available tools. You can take that to particular, you are interested disease oriented, like suppose you want to work in cancer or you want to work in some chikungunya or some kind of SARS-CoV or something like that. You get the sequence from this website, then you can construct the model. We will see the later. Then how you can construct the model, the protein structure, everything. So this is the website. You can get the information about the. So as I mentioned, as I told, so I needed to collect the sample because so each person may be a different kind of as I mentioned, 20% genome sequences is different. So we need to identify a drug that is called the personalized medicine. Personalized medicine. That means for me one sequence, for you one sequence. Once you go to the doctor will use like nowadays this is the everything stored in the ATM card. You go to the doctor, once you swab everything, that you will get the sequences about you. All sequences they will get. Then where you got the diseases, you can identify. That is the personalized medicine. Based on this affected place, they can give the personalized medicine for the student. So this kind of things, that is the purpose we need to collect the sample for each person and do the sequencing and get the information about the disease related oriented things. So like this way, there is a personalized medicine. So like you can see here, the protein, there is a drug molecule. That means ligand means drug molecule. There is a macromolecule. This is a small molecule. Then you can see the complexes. Once it is bind, it will start the mechanism of this. So we want this protein structure also. We want this small molecule structure also. So then we need the complex also. Then you can think about that. Here is the major role is playing with the physics. Okay. Next. So okay. So now we got the genome sequences of for a particular person. There is a million. There is like maybe like 6,000 proteins are there. 6,000 genes are there. So each gene do a different kind of function. Then we need to what kind of which protein is which function is doing. That is very important. Then we need to go for the analysis. So we use like many bioinformatics tools to identify each protein function. Then based on that only we can go for to design the drug or to propose the mechanism of the protein. So that is very important. So we can use next. So these are the tools. So these are the whole proteome retrieved from the NCBI. As I mentioned, Once we got the genome sequence, we will deposit in this site. So do all kind of things, you will get this kind of things. You can see here, from the water and really culture, you are getting the like sequence, amino acid sequence, nucleotide. Think about that. From the blood sample, we will get this kind of things. From there, we are doing all kind of analysis to identify the biological role and what kind of disease affected. So using this kind of all the tools, we can get everything, the protein family and what kind of domain and what kind of motif in the structure. So based on that, we can say about the function of the protein. Next. So these are the softwares, these are the tools. The MSc students maybe which is very useful for you. So maybe you can do your project work to like use this freely available all the tools. From take the genome sequence from the database, use these tools and you can get the all kind of information. Next. So as I mentioned, so I taken sample from one person. From the sample, there is a given a name for each person, one strain ID. They will give the strain ID. There is a strain ID. Then from there, this much of protein is there. You can see, think about that. This much of protein means gene. This much is a protein gene. Close to the 4,000. From there, there is a hypothetical protein. Hypothetical means there is an unknown function. We don't know. The remaining are already annotated. Maybe you can think about that more than 664 proteins are not unknown. That is means no function. So we needed to identify. Then from there, so we can get the like selected based on these informations we can collected, and finally we will get the there is a important proteins for 16. You can see here binding protein, how many proteins are there, and the transporter proteins, how many proteins are there, and the regulatory proteins and other proteins. These are enzymes. From this, there is a 6,000, the 3,839 proteins. We can get this kind of information. So based on this, we can go for the next level of studies. Next, okay. So now you got it everything. You know the genome sequencing, and you know the what kind of protein, enzyme, and transporter protein, and the disease related protein, everything. Okay, now you got the genome sequences. So once you got the genome sequences, now what we need to do? So there is a two methods are there. One is the computational methods, and another one is experimental methods. Okay, why need the computational methods? Why need the experimental method? As you know that experimental methods, we need a huge money and high sophisticated instrument and chemical time, etc., etc. Right? We need to spend a lot of money. Right? Computational, only one computer is enough. All the softwares are freely available. Then we can download the sequence. We can do everything. There is a two method, computational methods, and also. Why need a computational methods? We can do very fast because millions genome sequences from the various organisms deposited in the NCBI based on the various disease oriented. We needed to identify solve the structure of the protein. Then only we can go for the design the drug. That's why the computational studies emerged, the bioinformatics. Okay. Then next, there is a method like you can see here. There is a protein structure modeling and validation, then prediction, and do the all the molecular docking and the complexes. Complexes means drug is binding into the protein. Then study about everything. This is all everything involved the physics and the computer and biology, all. So combination of the physics, chemistry, biology, computer science, and mathematical science. That is the multidisciplinary area. So you can think about that. Physics person can do everything. Okay. So next. So as I told now, we are getting the one protein. There is a more than 4,000 proteins are there. I selected one particular protein which is related to some kind of disease, right? The one sequence I have selected, one gene. Okay, you can see the sequences. There is a 20 amino acid. The combination of 20 amino acids is making a one protein, right? It may be a different kind of length, 400 amino acid or 500 amino acid. Only 20 amino acid, combination of 20 amino acid. That is making a one gene. So here you can see here, there is one protein which is causing some disease. Okay. Now I am doing all kind of bioinformatics using the bioinformatics tools. Any other already reported structures or reported proteins are there, I need to check first. Then if it is not there, it maybe is there, maybe 30%, because 20 amino acids, the combination of 20, maybe it may be a similarity of 20% or 30% or 40%. So I taken this one, then I did everything, then I got this one. You can imagine, this is the protein structure. Through computational studies, using the computer, using the computational tools, you can see from the water, from the blood, I am getting the amino acid sequence. Then I am doing all kind of things. You can see here, whether you can able to see here structure from the water or from the blood, we can see the protein structure. You can see the structure of the protein. This is the macromolecule structure, that is called the protein. Okay. Then we need to do all kind of validation. Maybe this is our scientist. So unfortunately, he didn't get the Nobel Prize due to some, because he is from the Indian scientist. That's why he didn't get it, Ramachandran. So, but the entire world is using his method to validate the protein structures. There is a Ramachandran plot. Without this plot, they cannot do anything. That is the highly very important. So once validation everything, then we will go for the stabilization of the protein, because whether the protein is very stable or not, we need to check the various like molecular dynamics. So we need this kind of supercomputing facilities to do this kind of things. So okay, once done, next, then you can go for the check with the other reported structures, whether any other thing similarity with what you identified structure or protein, whether any kind of residues, that means amino acids interacting with the drug molecule. Those things we need to go for the study. You can see here, here you can see this is the protein. The each protein here is very important. Each protein there is an active site packet. That active site packet means, that means you are taking the drug, the drug will bind into the protein. That is the cavity. That is a packet. So once you are taking the drug, that drug will bind into the protein, then it will interact with the protein, then it will start the mechanism of everything. Because for example, you got your headache today morning before starting, right? So you want to Dolo 650, right? Then you are taken Dolo 650. Dolo 650, there is a structure. How many of you know about that? You are taking the tablet, now each tablet there is a structure is there. That is called a small molecule. That is called a drug. That is called a ligand. Do you know that a combination of what other things are there in the Dolo 60? Carbon, nitrogen, oxygen. So you know that. So each tablet there is a structure. There is a structure is there. It contains the carbon, nitrogen, oxygen, everything. There is a molecular formula is there. There is a molecular weight is there. The less than 500 weight we must use. Okay. So here you can see this is the protein. We got from the protein the gene. Then there is an active site packet. There is a small molecule. There is a drug molecule. You can see structure. This will bind here. Then it will start the mechanism. So once you taken this Dolo 650, after 1 hour you will reduce the headache. No? So that means what? It is started your mechanism. So slowly, that is the interaction between the protein and the drug molecule. For that we need the physics, X-ray. From X-ray we can solve the protein structure as well as the small molecule structure. Right? Okay, next. Then you can see here how it is interacting. There is a drug molecule, there is amino acid, how making the interactions. You can see here. Once interaction start, the mechanism will start. So this is the way working our all the drugs and everything, then cure our disease. Okay. Okay, next. So we do all kind of the analysis like talking and screening and dynamics using the computational like supercomputing facilities. Okay, next. Okay, now so we have collected a sample from the culture. We identify the genome sequencing. From the sequencing, we use the computer to solve the structure, to model the structure, right? Now over. So you can see, you can think about that. Biology from the water, genome sequencing, then protein using the computer method to identify the structure of the protein, everything. Now we want to do the experimental aspect. So what are the methods are there? Experimental. Yeah, a person, a physics person to work these much of things. You can see. What is the title? Cloning, expression, purification, crystallization, and structure determination of the particular gene or particular protein. So you can imagine. So here the role is next. So what is the method? So as I told you, you can see here how it is molecules. We want to determine the molecule. Computationally, we structure, we construct the structure. Now we are going to do the experimental aspect, biological. That's why I told you the multidisciplinary, like physics, chemistry, biology, chemistry, and then computer science, mathematical science, to involve everything. So next, so there is a two kind of things are there. This is a small molecule. That means drug molecule. Okay, small molecule crystallography. That is small molecule crystallography. Maybe you studied about in the solid physics third year, like a crystal system, monoclinic, triclinic, orthorhombic, or something, and the space group P21, P2, something symmetry related things. There is a small molecule and a macromolecule. Small molecule means the less than molecular weight 500. Macromolecule means, as mentioned, that's amino acid sequences. Protein, drug molecule. There are the two kind of crystals we need to make. There is a single crystal. That is a very important, not for powder. Single crystal with periodical arrangement of atoms or molecule. That is very, very important. So not like powder sample, right? Okay. So this is the next. So this is the methods to solve the structure. So you can see here X-ray crystallography. What is this? X-ray. X-ray is coming from where? Physics or biology? Physics. What is this? Diffraction pattern, right? From where we are getting this diffraction pattern? We are using the X-ray technology. From the cells, what is things are there? Atoms, molecules are there. Na? The atoms, what it contains? Electrons, protons, neutrons. From there we are getting the photons, diffraction pattern, right? This is the method. These methods, everything solution oriented, but this is the ultimate method to solve the protein structure. This is a very, very important. So from here only we can say about exactly, accurately to say about this molecule, this atom is interacting with this amino acid or this atom, like that. That is very, very important. Here is the solution study. These many of studies are there to solve the protein structure. Okay, next. So this is the method you can see here. So I told you na, there is a one sequence now taken na. One sequence. There is many kind of sequences are there, but particularly you are targeting one protein that is related to some diabetes or some chikungunya. You need that protein structure. Then only you can design for the drug for that particular disease, right? So we need to target. Then we need to go for this kind of construction. Then we need to transformation. This is everything biology. Then we are going for the expression to check about whether your gene expressed or not. Then we will go for the large scale. Here is the very, very important. Why you need a large scale? We need a large volume of protein to make a crystal, single crystal. There is a n number of methods are there. So to make a crystal, screening also is there. Until to get a crystal, you cannot say about that. You cannot able to solve your protein structure. It may be, sometimes it may be like salt crystal also. So for that, we need a large volume of protein to go for the crystallization method. So no. So once you got and use the all kind of instrument to break your cells and then protein purification, then here you can see here my targeted protein. Many proteins are there. Here I can see from this using these methods this is my protein to express. From there, I need to go for the crystallization. There is a many kind of methods crystallization is there to make a single crystal to use it. Finally, you are getting the crystal, single crystal. To take this crystal to use the X-ray to get the data. Right? Next. So these kind of facilities are needed for the to making the crystal. You can see this culture and centrifuge and the purification methods. It's very important. Why I am telling this one? Only computer used to constructing model, right? Only one computer. Here you can think about that how much is needed for the experimental aspect. So that's why we are lacking. See, when it was Watson Crick, when he was solved the structure, 1965 or when Watson Crick. So you can think about that up to now only 2 lakhs 20 protein structures available from not only human, from various organisms. So you can think how many years, almost 80 years. 80 years only 2 lakhs 50,000 structures from also various organs. So you can think about that. In human proteins only 15-20% proteins only solved. You can think then again 20%. As I told you, there is a 23,000 proteins are there in each cell. Only 20% solved. Then we are lacking for the medicine for many kind of diseases. That's why we cannot be able to cure many diseases exactly. We are using the already identify the drug. So using the analog drugs for this combination of drugs. Nowadays we can go to the doctor, they are giving the 3 tablets together, right? 3 tablets together, cocktail or something oriented to cure the disease because already we got the resistance. So that is not accepting medicine. That is also one big problem is there. Already existing antibiotics not accepting the our disease that medicine. So that's why the doctors are going together, three tablets together, 4 tablets are together. So sorry. So this kind of facilities needed for the experimental aspect. Next. So you can think that, so as I told you, this is like we are getting the sequencing already deposited from the culture. We are taken this one, we got it, and we are doing this kind of things and doing the all biological oriented, and we can check the DNA and then culture and your protein expression method. This all biological oriented. So once we identify this protein, next, then we will do all kind of this kind of purification to purify because we need a purified protein to go for the single crystal because many proteins are there. Our target one particular protein to purify everything. Then you are checking the molecular weight of the protein. Each protein there is amino acid, carbon, nitrogen atoms are there. Each atom there is a molecular weight is there. Then we need to go for the molecular weight, check whether I targeted protein is exactly matching or not. So to identify this kind of methods. So once you got this one, next, then we have the n number of crystallization methods are there to grow a single crystal. So that's why it will be very expensive. So you can see that many crystallization conditions until to get a crystal, we have to go for screening. If you failed, then again you need to go for to change your construction, to change your purification method and everything. You can imagine how much money we need, how much time is needed, how much we have to spend everything. So that is very important for until to get a crystal. There is many methods also is there. Using one method, using the 4,000 conditions. You can imagine 1 microliter protein, 1 microliter buffer condition. Like what you are making the crystal, like powder sample preparation and everything. Then use the 4,000 condition to check whether its crystal is grown or not. If not grown, then you have to change the construction methods. You have to target other way. Then change the method, change the buffer, change the methods to grow the crystal, sitting drop, hanging drop, micro batch method. Then many methods are there. Imagine 4,000 conditions are there. Then how much protein is we need, how much buffers are needed to grow the crystal once you are failure. So this is a very tough and very difficult job. We need a huge money. Then once you use these things, so like next. So we are getting this kind of conditions, getting the crystal. You can see the crystal. See, just imagine from the water, from the sample, from the blood, we are getting what? Crystal. So just imagine, think how this everything is playing the role. So once we got the good crystal, then once got crystal, where we need to go? Then we need to test whether it is a crystal or not, whether we are getting the diffraction or not. That is very important. Next, then we are using the diffractometer, X-ray diffractometer. Maybe you know about the like cyclotron, synchrotron, something like that. So use this kind of diffractometer to check whether your crystal is diffracting or not, until to get the diffraction pattern. If not getting, maybe that is a star salt. Then again we need to go the beginning. Then do everything. Then we need here also very good resolution is very important. Why need a resolution? Because the protein and the drug molecule or ligand molecule interaction. There is a hydrogen bond interaction, CHO hydrogen bond interaction, NHO hydrogen bond interaction. Like one was portion, the hydrogen bond interaction. Because then only the electron move from here to there, then only mechanism will start. So we need to exactly to locate the which is carbon, this is hydrogen, this is nitrogen. Based on the interaction, we can say about that. For that, we need a high resolution data. That is like Armstrong. You know what is Armstrong? 1.5 Armstrong means what is the unit? 10 power minus 10. Just imagine 10 power minus 10. So that kind of data is needed for to solve the protein structure. Okay, next. Okay. So this is very important because the crystal what you are taking outside, maybe it won't live for, that is not to stay for a long time. Then we need to collect data immediately. Then what kind of technology is needed? That is a synchrotron. That is a synchrotron facility. So you know the synchrotron? You studied maybe synchrotron. Like high power. You can see that like this is a synchrotron facility like springite, super photon ring, 8 giga electron volt, 8 giga. You just imagine 8 giga. Giga means how much? That much of power. You can imagine just crystal, only very size of the crystal might be 0.3 millimeter crystal. We are passing the 8 giga electron volt to get the diffraction data. So we are using this kind of synchrotron facilities to quickly get the data because otherwise crystal won't stay very long. Maybe within 10 minutes or 1 hour it will be. That's why in-house diffractometer, it takes more long time to get a data, 3 hours, 4 hours or something. But if you use the synchrotron facilities, maybe you can get a data within 10 minutes, 360-degree angle. 360-degree angle you can get the data. Then once you got data, then we will go for the, like this is the super photon ring. Nowadays, so we are making a crystal, it is a very long process. So once you purify the protein, you will get the protein solution. That is XFEL, that is a X-ray free electron laser. So once you give the one drop of the protein, you will get the data, image data. From the data, you can get the structure of the protein. So this is the way going on. But again, we need to come to the single crystal. That is a very, because there we can get the only the whole protein, everything. So this is the fastest method for to get the data to go for the solve the protein structure. Next. So here you can see here. So we got the solution protein. So now we got the protein solution. Then you are using the various methods to get the, you can see here the diffraction. Then you are getting from the diffraction what are things we can get? Cell parameter, right? ABC and angle, alpha, beta, whether belongs to monoclinic or triclinic or orthorhombic or tetragonal, something we need to specify the space group of the system, right? Then we are using all kind of techniques to solve the, get the data, raw data. It will give the plane, Miller in this plane, each plane. 2D sin theta equal to n lambda, right? Plane is there, then 111 HKL, right? Miller in this 111, 211 per day. Each plane you will get the intensity. The once X-ray diffraction will come out the diffraction pattern, that is you will get the intensity. So you can imagine how many planes are there, how many intensity will get. Then you take all data to analyze to solve the protein structure. You can see how many, whether it is possible to count this diffraction pattern. Each will give one intensity, one plane, 111, maybe 1500 intensity or something. Here there is a Fourier transform, structure factor, electron density calculation, right? Fourier transform, maybe you know. So from there we are calculating the electron density map using this data. Okay, next. So these are the methods are there to solve the protein structure. Maybe everything related to physics. So like molecular replacement and maybe SIR, SAD, MAD, anomalous scattering, single anomalous scattering, multiple anomalous scattering. Like these methods are there to solve the protein structure. Okay, next. So here you can see here. So just imagine from the solution getting crystal, then you can see the electron density map for the each amino acid. There is a 20 amino acid, combination of 20 amino acid. You can see these are all amino acids. These are all electron density getting from the structure factor, Fourier transform, right? Electron density. Based on this, we are solving the structure. We are collecting data. We are getting this kind of electron density map. Next. So you can see here, as I told you, what is this? What kind of diffractometer you are using? What kind of wavelength you are using? There is a two kind of wavelengths are there, like 3 or something like that, like copper K alpha radiation, molybdenum K alpha radiation. If you are going to use the springite, then you can vary the energy to get the data based on the resolution. Okay, you can see here everything related to physics are right. You can see space group, cell parameter, resolution, and everything is related to physics. Okay, what is this? This is the native form. Any structure, there is a protein structure now we got, right? Any structure without any ligand, that is a native structure. See, once you have the structure without any ligand, what is the use? No use, no? Only. That is called the native structure. We needed to find the first native structure. Then we have to do all kind of these things. Once the native structure solved, right? Then again I need to go for to solve the complex structure. Complex means drug molecule. I have to do the purification. I have to make a crystal. I have to collect the data to solve the structure to get this data. This is a protein, then complex together. Then many kind of substrates are there. Then many kind of amino acids are there. Mutation. Like mutations are there, maybe during the COVID time, they told about that there is a number of mutation, like first initially SARS-CoV or something. Then finally it came alpha, beta, gamma, something it came, that is a mutation of the amino acid. So I need also solve the mutation structure also. You can imagine how many structures we need to solve to propose the mechanism. Then you can see this like native data, complex data, then another one complex data. Same protocol I need to do in the experimental. Here I want to tell these things. In computational, you can take foot, that's all enough. But the experimental, we need to prove everything from the data, right? Okay, next. Then you can see here. So I told you, there is a native structure. Only solve the structure. So no use. We solve the structure. But we cannot say about this protein belongs to this function, that's all. So here you can see the drug molecule, right? The drug molecule bind into the protein. There is an active site pocket. Each protein there is an active site pocket. You can see the drug molecule bind. Then another drug molecule. These are the complex structures. So based on this, we can propose the mechanism how this protein is working and everything, right? So this is the way we can go for the experimental studies. Next. So here you can see the electron density of the drug molecule. Maybe you can think $650. This is a $650 bind into the protein. Once you are taking medicine, It will go and bind to the protein. So this is the way we needed to identify the electron density observed and calculated. Structure factor, maybe equation is there now. From there you can get. Okay, next. So you can see the mechanism of the protein. Is a protein structure, is a protein. There is a amino acid, then a ligand molecule. There is some ATP molecule bind into the active site. Here you can see what kind of things are we are getting. This is a reaction mechanism. So we need to propose reaction mechanism. So that is a, so ATP means this is an energy giving molecule. So once we take the food and everything, we will get the energy. Na? That is ATP. ATP won't bind. It will come out of the ATP or pyrophosphate like this way. So this is the mechanism we are proposing. Next. Then after that we need to make a tRNA because this protein is belongs to the amino acid synthesis protein. So then we are making a tRNA. Then we make a complex of this protein with a small molecule and then tRNA. Then it is working the charged tRNA. It is giving the amino acid synthesis in amino acid in our body. So this is the way we need to go for the to solve the protein structure and propose the mechanism. Next. So you can see here how it is binding. This is a protein, right? The protein, whatever you have taken $650, it is binding into the active site pocket, then it will start the mechanism. Then we need a two structure. One macromolecule structure need small molecule structure. That means drug is a macromolecule bind. So we have to solve these structures, then propose the mechanism. Right, next. Okay. So as I mentioned, you can see here this image. So there is organs, various organs. Each gene there is a protein. So you can see how many proteins are there. Just for example, I am telling. In one organ you can take 700 genes or 700 proteins. So whether you can identify exactly what disease causing this one, that is a problem. So we need to solve like protein structure. You can see if you target the one disease, one particular cell, one gene from our body, how long it takes. You can target, then validation we need to do, and to identify some drug molecule. You can do entire process to identify the drug development. It takes 50 to 20 years, 15 to 20 years to come out one drug. Then you can imagine whether you can be able to solve, we can find a drug for the immediate for diseases. So this is the very long process are there. So entire world is working on this aspect to identify. Each scientist working, biology science working on this way, material science people are working that way, and sensor people are working that way. So everyone is working together to bring a technology and to identify everything. So you can imagine this much of efforts is needed to bring a one drug molecule. Next. So you can see here how we are getting. There is you can take any kind of plant, any plant. There is a medicinal plant they are telling now. Maybe now we are taking some kirai na. You can take that kirai, maybe many like molecules are there. Drug molecules are there. So once you give the GCMS, you will get the each from the leaf you will get the 30 molecules, 40 molecules. There we need to identify one molecule which is causing, which is curing the diseases. That is very important. Then we have to go for the all kind of screening and everything. Number of molecules, then we will go for the preclinical studies, clinical trials. Finally, we will get this molecule. You can think how many years are taking to bring out one drug. So maybe you think about that, today you take this kind of kirai, then it will cure these things. Tomorrow you take, every day you take that kirai leaf, you take the leaf, how many molecules are there? Do you know? One leaf. So maybe each leaf there is a 40 or 60 molecules are there, small molecule. We don't know which molecules are taking for this. You take carrot, eye is very good. In the carrot, how many molecules are there? Any one molecule, maybe it will cure that one. Then if you continuously you take, it will cause also effect. If you take the daily kirai, it will cause something because acidic molecules are there, good molecules are there, bad molecules also there. That is very, very important. So we need to also give much attention. Also, those things also very important. It will cause other kind of problem. So like that, we need to identify the particular molecule, particular diseases. So that is next. So you can see here, a person identify the diseases, then how long it takes, then we will go for the preclinical studies, then we need to test the animal model, then we need to scale up, because we need to scale up. $1.60 is not enough for one person, because millions of populations are there. Maybe today in Tamil Nadu itself, maybe 3 lakhs person got a headache. Okay, we need a 3 lakhs $650. Then we need to scale up. Then we need to go for the synthetic aspect. That is a chemistry is playing here. We have to go for synthesis of that particular drug. Right? So once they are synthesized, then we will get the powder sample. That powder sample we need to make a single crystal. Right? Single crystal. From the single crystal only we will get the data, X-ray data, to solve that single structure also. The small molecule structure also we need to solve using the crystallography method to solve the structure. Then this is the way. So we can think about that, how long it takes to come out the one structure and protein and everything. Next. So you can see here there is a experimental method, structure biology, as I explained from the solution to the protein structure biology. Here from the plant, we can take the plant, any kind of medicinal plant, you can get the drug molecule. Like marine source, marine compound they are telling, plant compound they are telling. From there we can get this kind of drug molecule. We need to identify, we need to screen and everything. So we will get everything. Then use the computer technology. As I mentioned, from the sequencing we can go for the computer oriented. That is computer method, everything is only predicted method, not exact method. Predicted method means it may be. Exactly we cannot say about this drug to this one. But the experimental only, it is an exact method. That's why we can go for the experimental aspect. This I told pharmacogenomics. Nowadays it is coming a pharmacogenomics. Why need a pharmacogenomics? Personalized medicine. So now the country is working on to bring a personalized medicine for particular diseases. To go for to get a sample from your body, get a sequencing and store in the ATM card. Then go to the doctor, they will give the complete sequence about your history, and particular segment is based on this segment, you got this kind of diseases. For that, I am giving this medicine, because you can think about that. There is a doctor is giving the 10 days medicine. One medicine is 500 mg. So at a time, the morning you need to take a 4 medicines. 500 mg into 4, how much? 2000 mg, right? 10 days. 3 times, this is 3 times 6,000. 10 days. Just imagine how much drug you have taken. That will cause any kind of disease or not, side effect. Right? So that's why we need to keep much attention for our health aspect. And nowadays, the scientists are working to identify the nano drug. Nano means how much? 10 power minus 9. You can imagine. Mg is there. This is a nano. 0.00001 gram only we are going. That's why nano drug delivery system is coming on the way. So for that we need the personalized medicine. Personalized medicine. So this is the way it's working the entire world to identify the drugs for everything. Next. So like that, so you can see here, this is a biology person to involve, biology person, that is physics person to get the diffraction data and everything, and a chemistry person to synthesize the molecule, right? To synthesize, to solve the small molecule we need the chemistry person drug. So here you can see how many people are involved in, right? Then you can see marine sources. There is a n number of resources are available in the sea to get the marine compound to make a drug for everything. Then you can see the biochemistry person is checking everything, clinical oriented aspect, do all molecular biology work, cloning, expression and purification doing by the biological person. And you can see bioinformatics person to involving to identify the sequence aspect and everything. So here you can see entire all people are involving, sensor people are involving to identify sensors. Immediately we are getting result. Na? Once we given something, so you have this kind of things like sensor related, biosensor. So all people are involving to bring this multidisciplinary teaching and research. So you can any person to work on the any area to together to bring out the technology as well as the medicinal aspect. Next. So for this kind of things, we need this kind of facilities which is available in our department. So maybe if you are interested, you can come for the summer project to do all kind of work, and also faculty also can come and to get the experience how to do the purification, crystallization, and using the bioinformatics tool and get to know and everything. Next, so and our department is offering this MSc program and MPhil and PhD programs. Those who are interested, you can come and join in our department PhD. Next. So these are all my scholars. They are all working in different areas. Like you can see cancer and Acinetobacter and there is pathogens related, escape pathogen. So all are working and all are postdoctoral fellows working in the different areas. Next. So these are the fundings. So I got the various funding agency to set up the lab and everything. This much of funds supported more than 25 crores I received up to now. So next, this is our science block, only science block, which is located in the 4th floor of my department. So this is the way we need to work to go for the computational as well as the experimental aspect. So thank you. Any question? Maybe you can. Okay, madam. Time is. கண்டிப்பா முடியும். There's way personalized something is. உங்களுக்கு எடுத்து உங்களுடைய ஜீனோம் சீக்வென்ஸ் பார்க்கலாம். மெடிசன் கொடுக்கணும்னா அப்படிதான் பண்ணணும். ஆமா. ஒவ்வொரு ஜீன் எடுத்துட்டு அதுக்கு தக்கன பண்ணலாம் நம்ம. So now it's going on, but it takes a very long time. Personalized medicine, it is a pharmacogenomics related things. Yes, sir. Single crystal model. So we can go for the solution, but it will give the overall structure. But single crystal only exactly to say about the reaction of the ligand and protein. Nanotechnology also. So we need a single crystal. So without single crystal, we can't say about that. So, anything? So maybe maybe the student, MSc student or UG students, maybe you can. No need to think about I have taken physics, maybe I didn't taken this biology, I didn't taken chemistry. So we can together play everything. So
when we take carrot or green leaves, there is no side effect.
no, it won't cause side effect if you take more. Because if you take carrot, maybe 40 molecules are there. Okay, that 40 molecules you need to categorize. Whether how many molecules good molecule, how many molecules bad molecule. So if you take regular daily, like 1 hour, you can take one carrot, next 1 hour, like this way, it will cause some problem. Na? Because molecule, everything molecule. If you also take any கீரை, பிளான்ட்ல எவ்வளவு மாலிகுல் இருக்குன்னு தெரியாது. சும்மா நம்ம சொல்றோம் இந்த கீரை சாப்பிட்டா நல்லா இருக்குன்னு. அந்த கீரையில் 40 மாலிகுல் இருக்கு. அப்போ வேரிய ஸ்டடிஸ் பண்ணி அந்த 40 மாலிகுல் எடுத்து செக் பண்ணி பார்த்தோம்னா அது அசிடிக் இருக்கும், அசிடிக் மாலிகுல் இருக்கும். அந்த மாலிகுல்லாம் ஒதுக்கி விட்டுட்டு எந்த மாலிகுல் குட் மாலிகுலோ அந்த மாலிகுல் தான் இட் வில் கோ அண்ட் பாயிண்ட். அப்போ நீங்க அசிடிக் சாப்பிட உங்களுக்கு ப்ராப்ளம் தானே வரும். ஸோ இட் வில் காஸ். அதனால எதுவுமே லிமிட் தான், ஒரு சடன் லிமிட். ஸோ எவ்ரி திங்ஸ் ஆர் தேர். ஸோ
500 mg கீழ நீங்க அதுதான் எடுக்கணும்னு சொல்றீங்க.
அப்படி இல்லை, ஒரு எக்ஸாம்பிளுக்காக சொன்னேன். அதாவது நீங்க டேப்லெட் எடுக்க எவ்வளவு ட்ரக் நீங்க சாப்பிட்றீங்க, அந்த ட்ரக் மூலமா இட் வில் கோ டெபாசிட் எவ்ரிவேர்ல. அப்போ அங்கும்போது it will cause other kind of disease, other kind of problem will cause. Suppose you are taking for medicine for diabetics, diabetic medicine எடுத்தவங்களுக்கு என்ன வருது, திரும்ப ஹார்ட் அட்டாக் வருது. தட் மீன்ஸ் பிளாக்ல ஏதோ அடைக்குது சம்திங் இல்லையா? ஸோ அந்த மாதிரி தான். அதனால வந்து ட்ரக் எடுக்கிறத வந்து அவாய்ட் பண்ணணும்னு சொல்றேன். தட் இஸ் வெரி இம்பார்ட்டன்ட். ஹவு யூ கேன் அவாய்ட் தட்னா, யூ நீட் டு ப்ரீகாஷன் எவ்ரி திங், டூ எக்ஸசைஸ், டூ சம் யோகா அண்ட் ஜங்க் ஃப்ரூட்ஸ், ஜங்க் ஃபுட்ஸ், அதாவது இந்த ஆயில் ஓரியன்டு இந்த மாதிரி ஃப்ரைட் ரைஸ் இதெல்லாம் வந்து கொஞ்சம் அவாய்ட் பண்ணணும். ஸோ அந்த மாதிரி இருந்தால் தான் வி கேன் சஸ்டைனபிள் அவர் ஹெல்த் அண்ட் எவ்ரி திங். ஸோ and water also very important. Contamination very important. So
good morning, sir.
yes ma'am.
It was very useful today. As ரெண்டு மூணு டவுட்ஸ் இருக்கு சார். ஸோ சில பேருக்கு வந்து சில மெடிசன்ஸ் எடுத்தாங்க அப்படின்னா இட்ஸ் பிகமிங் லைக் அலர்ஜி. வாட் ஐ ஜஸ்ட் சா அது வந்து இப்போ நீங்க காட்டுறதுனா ப்ரோட்டீன் வரதுனாலயா இல்லைன்னா இந்த பிளட்னாலயா இல்லைன்னா சம்திங் ப்ரோட்டீன்ஸ் ஆர் டிஎன்ஏ.
சப்போஸ் அந்த ட்ரக் வந்து அந்த புரோட்டீன் போய் ப்ராப்பரா இதாகல, இல்ல அதனால வந்து உங்களுக்கு வேற ஒரு சீக்வன்ஸா கூட இருக்கலாம். அந்த சீக்வன்ஸ் எடுத்து பாத்தீங்கன்னா இந்த 20 அமினோ ஆசிட் லைக் அலனைன், இது டெஃபடோபான், லூசின், ஐசோலின், there is a 20 amino acid. The 20 amino acid there is a essential, non-essential is there. Some amino acids our body will synthesize. So 8 amino acids or something. So other amino acids we are getting from the food. You can see from the food it will change everything. So maybe you take the your genome sequences, it compare with the other person. It may be some amino acid may be differ. Instead of alanine, maybe losing for you. So that drug will go and bind there. Maybe there is no interaction. That's why you cannot cure your diseases. So that purpose. It may be. That is, we need a in-depth studies. But this is like this kind of things. Mutation, it will happen. There's the genome sequences. Each person maybe.
And few things that all I hear, like the painkillers, like Zintac, Rantax, and acidity problems. They used to say kidney will fail, don't take.
That's why I'm telling you. So if you take more medicine, it will cause the side effect.
Everything it will
Everything. Any tablet you take, it will cause the side effect.
And the crystal சொன்னீங்க சார், அது ஒரே ஒரு டவுட் தான். அப்ப அந்த கிறிஸ்டல்ல வரும்போது we can see the exact shape of that virus or
no. From the crystal there is I told you the 20 combination of amino acid.
Okay.
That is the structure. We will get that is I told you the molecule contains what? Atoms. This is a blood sample, tissues contains of the cells contains of molecules and atoms. இந்த மாதிரி தான். So yes ma'am.
Recent problem. That is the tablet pop inhaler is better. Small amount of
அந்த அளவுக்குலாம் என்னால சொல்ல முடியாது. சயின்டிஃபிக்கா இது மாதிரி எல்லாம் வரும்னு சொல்லலாம். ஏன்னா these things are we need to discuss with the doctor madam. So because this everything is medicinal oriented. How it is functioning, everything we can talk. How it will cause the problem, we can talk. So how it is working, what kind of medicine is working, depends upon each person. See, this SARS-CoV during the SARS-CoV time they told, the mutation is occurring how. So for me, it's a normal COVID. Once everything spread from others, it will change some other COVID. Alpha, beta, gamma. That means mutation has happened that your body itself. So my body is normal. Then once it is from my body to other body, something mutation happens, then mutation coming as a other virus. That kind of things. So it depends upon each person biological system. So
the medicine எப்படி அந்த நோயை குணப்படுத்துதுன்னா அது காமனா
இந்த மாதிரி தான். ஸோ ஒன்ஸ் யூ டேக்கன் மெடிசன்னா நீங்கள் இப்போ அதான் சொன்னேன் இல்லை, தலைவலிக்கும் போது நீங்கள் டோலர் 650 எடுக்கிறீங்க. டோலர் 650 எடுத்த உடனே வித்தின் 1 hour வந்து உங்களுக்கு ஹெட்டேக் ஓடி போகுது. எப்படி? தட் மீன்ஸ் த ட்ரக் மாலிகுல் பைண்ட் இன் த பர்டிகுலர் ப்ரோட்டீன். ஸோ மெக்கானிசம் ஸ்டார்டட் ஒன்லி இட் இஸ் கிராஜுவலி டிக்ரீசிங் எவ்ரி திங். ஸோ திஸ் இஸ் த வே எவ்ரி திங் இஸ் ஒர்க்கிங். We cannot say inside how it is working.
We have common cough for everyone. But the tonic they take.
That is it depends upon our taking food and water and everything. In the food,
It's common. But ஒரு medical
there is a one kind of flu. There is a virus. That is a,
tonic keeps on changing.
hmm.
ஒவ்வொரு ஷாப்ல ஒவ்வொரு tonic.
No. If you check the structure of this everything, maybe everything similar. Maybe small kind of maybe altered, maybe the analog
I
oriented.
have experienced one thing. Third standard kid, he has a fever, shivering fever, morning summer. So he went with his parent. And the doctor வந்து they gave the medicine. அதில் வந்து டோலோ 350 யூஸ் பண்ணி பேராசிட்டமால் டேப்லெட் கொடுத்தாங்க. நீங்கள் சொன்ன பாயிண்ட் ரைட், ரிசர்ச் பண்ணி பண்ணால் என்ன ப்ராப்ளம் சால்வ் பண்ணலாம். பட் தேர்ட் ஸ்டாண்டர்ட் கிட்டுக்கு டோலோ டேப்லெட் வந்து ஜாஸ்தி தான். பட் பேரண்ட் அவேர்னஸ் இருந்தால் மேபி கேட்பாங்க.
நீங்க அந்த டயத்துல வந்து நீங்க கொஞ்சம் மினிமம் கொடுத்து பார்க்கணும். அந்த மாதிரி
நமக்கு
அது வந்து எதுவுமே நம்ம சொல்ல முடியாது. So it will be depends upon the body, but how it is working the mechanism, how it working the mechanism, what kind of things we can get it from the our studies. So அந்த மாதிரி சொல்லலாம். So it's need a lot of things to do everything. So
அதனால பாயிண்ட் ரிசர்ச் பண்ணி பண்ணாலும் கரெக்ட் தான்.
that is going on. It's almost more than 70% we are the achieved everything. The remaining 30% we need to find the exact oriented. So as I told you, there is a human protein only 23,000 protein இருக்கு. அதில் only வந்து 15% தான் பண்ணியிருக்கு. அப்படிங்கும்போது அதர் டிசீஸுக்கு எல்லாம் டேப்லெட்ஸ் எங்கே போகிறது, அதான் கம்பைன் பண்ணி கொடுக்குறாங்க. ஸோ அப்போ கொடுக்குற டேப்லெட் இப்போ ரெசிஸ்டன்ஸ் ஆயிடுச்சு. இப்போ வந்து 20 வருஷத்துக்கு முன்னாடி பண்ண வந்து ஆன்டிபயோட்டிக் ட்ரக்கு இப்போ சாப்பிட்டோம்னா அது வந்து ஏற்றுக்கிற மாட்டேது. காரணம் என்ன, அவர் பாடி ஆல்ரெடி ரெசிஸ்டன்ட் ஆகி தூக்கி எரியுது, வெளியில் தள்ளுது, எல்லாம் பிரேக் பண்ணி விட்டுருது. அப்போ என்ன பண்ணுறாங்கன்னா 3 மாத்திரை சேர்த்து கொடுக்குறாங்க. அப்போ 3 மாத்திரை 4 மாத்திரை கொடுக்க என்ன ஆகும், அவர் பாடி side effects, these kind of things will cause. So we have to identify the exact tablet for the exact disease. So for that, this kind of studies we need to do. There is once you want to do protein, you need to purify the protein. Once purify the protein, that is only your protein. So n number of proteins are there in the cell, but you are targeting particular protein, particular gene. So once you purify that protein, then you take that protein, do the crystallization.
Know that what are preparation methods are coming.
This is everything biological related things.
So can we do in the room temperature also?
So we'll
We can do room temperature, we can do in the -4 degree, everything we can do. Depends upon the your stable of the protein. So, like maybe you can try for the chemistry people synthesis, they will give the powder sample. You can use the powder sample to make a crystal. How? There is a methanol, ethanol, chloroform, acetone to make a crystal. You will get a single crystal. Then take that crystal, do the diffraction, X-ray data to get the structure to use the powder.
What kind of materials are preparation?
There is a number of things are there. We cannot. There is a very like see.
And she is a single crystal person. She grows crystals. And there'll be a lot of people.